Okay, now let's talk about something called developmental neurosimulation and interference-diffusion pattern formation. So this is work that crosses our representational brains and phenotypes interest group, our phasic group, which is physical intelligence, augmented systems and complexity, and our computational developmental systems interest group. So it's a wide range of different things going on here, very interdisciplinary. And I have a video on interference-diffusion pattern formation on the DevoWorm YouTube channel, so you can check that out at your leisure. So we're talking about something called interference-diffusion morphogenesis. These are large-scale interference patterns that are sometimes also called Moire patterns. And you can see in the animated GIF on the left what those look like. They're produced when a partially opaque ruled pattern with transparent gaps is overlaid on another similar pattern. So say you have two striped surfaces and they overlap with some offset. If you move them against one another, they form these Moire patterns, which are of interest to us. These two patterns must not be completely identical, so they have to be displaced or rotated or have a slightly different pitch. We'll see examples of this in the lecture here, but for now, let's just look at this pattern and look at what we have here. We have these two patterns that are overlapping, that are moving against one another. Sometimes it's motion, it's driven by motion. Sometimes it's driven by some sort of displacement. And there's this visual illusion of this complex pattern. So Moire morphogenesis is this form of pattern formation, this active pattern formation from growth processes and from these overlapping morphologies that kind of align with one another or unalign with one another. So we have a couple of components. We have a source component. So in morphogenesis, often we have sources of some sort of chemical or some sort of material that's growing outward from the source. We have diffusion from the source. So a source often produces something that diffuses outward. And in a diffusion process, we often have periodic or normally distributed decay or diffusion, which goes outward in every direction equally. Sometimes we don't have that. Sometimes we have some sort of heterogeneous diffusion or some sort of uneven diffusion, and so we can have that as well, and we can create patterns like we saw in the previous GIF. But in this case, we have diffusion going in all directions equally at an equal rate, and so those diffusion patterns form fringes, which are the set of concentric circles. So imagine in each one of these concentric circles, you have this gradient or this concentration of the diffusive substance. If from these fringes, then we get these gratings. So say we have multiple sources in space, and there's diffusion happening from each of these sources, and fringes are being formed. And those fringes will ultimately overlap and form these interference gratings, or these interference patterns. And the interference patterns from these gratings that you can see here, that they start to form these more complex patterns. And then you also have ripples, where you have this active movement, one gradient against another gradient, and they form ripples. So they have all these different properties of these basic components, these building blocks of Moire morphogenesis. So there are two features of Moire patterns that we're interested in. The first is relative motion, which means that they're these small movements that result in large-scale changes in patterns. And these result in these emergent optical phenomena. But as we'll talk about, these aren't just optical phenomena, they are optical plus mechanical phenomena. And so they appear to be visual illusions, but they have properties that are interesting to material science and ultimately to morphogenesis. So you have relative motion, you have this motion from a source, you have motion in terms of these ripples, you have motion between layers. And then you have this misalignment, which is the other component. You can't have them fully aligned because they'll just form sort of a reinforcement of the pattern. They need to be offset in some way. So this is where we have the same patterns. We could have two of these circles with the source has concentric circles and all of that, but they're offset in space so that they're misaligned. So that you can have the same pattern of concentric circles with this phase angle offset. Where in the case I just showed you, it's a two-dimensional surface. They're both kind of on the same plane, so they're offset by a distance. So you have this n-fold asymmetry, and this will become important in some of the examples we'll see later. We have these connections between wavefronts and a bilayer. So imagine we have an overlayer and a sublayer or a substrate, and we have interactions between that overlayer and the substrate or the sublayer. And those interactions then form this potential for pattern formation because the two layers are misaligned and they can move relative to one another. And you can get these kind of complex patterns from what is essentially a duplication event of the same pattern. So we can measure this in a number of ways. In the physics literature, in the material science literature, they use things called twist angles or analysis via density functional theory, or DFT. So there are a number of ways you can measure these kinds of pattern formations. And we're not going to get too deeply into the measures, but we're going, in fact, I talk about some of the measures in the talk on the DevoWorm channel. But for now, let's just keep in mind that they can be measured in pretty rigorous ways. So this is the video on the DevoWorm YouTube channel. It's called Interactive Morphogenesis, Sources and Dynamics of Moire Pattern Formation, in which I go over a couple of models of interactive morphogenesis. We have compartmented models that form these kind of simple examples. And so this is a longer discussion on morphogenetics in phenotypes and soft active materials, and it's that link. So this is a demo of Moire patterns. These are two concentric circles that are overlapping. So in this case, they have these transparencies that they have that are overlapping, and there's relative motion where you can see that the transparencies are being moved against one another, and you have this pattern evolve where you have these concentric circles that are forming even denser fringes against each other. So in Moire patterns, imperfect alignments can lead to interesting patterns, and this is a demo from the Exploratorium. This is an example of a demo that's built in processing. This is Clinton McKay's work on Moire pattern generation, where he uses a fairly simple, fairly small amount of code to generate these complex Moire patterns. Now let's review some of the work that we've done on developmental computational agents and how they acquire morphogenesis and, by extension, acquire behavior. This has been talked about in our presentations at the Embodied Intelligence Workshop in both 2024 and 2025, and those videos are also on our YouTube channel. So go look for our Embodied Intelligence Workshop playlist and you'll find more information about it. So a summary of that work is that agent embryogenesis proceeds as a series of morphogenetic movements or acquisitions. And so this is an example where you go from this sort of tripoblastic precursor to a situation where each one of these layers differentiates into different components of a vehicle. So in this case, we have sensors and effectors with a central nervous system, and we have these movements of the different layers of the precursor where you have the series of inductions and differentiations that form the phenotype. So eventually you'll get different agent phenotypes from this based on the different morphogenetic acquisitions or differentiations, inductions, etc. that you see. So it's agent embryogenesis proceeds as a series of morphogenetic movements or acquisitions. This actually also allows for developmental computation because we have an underlying artificial genome that we use to partially determine how these things develop. But we also use evolutionary approaches, and we can compute how this morphology can help in later cognition, how it sets up cognition, and how cognition then can be executed on these phenotypes. And so we can take these layers and we can have these induction events, these differentiation events. We can also divide up these layers as a series of discrete cells. So this is a case where we have these discrete cells, and these discrete cells deform along with these layers, and they also perform these specific developmental computations. So they divide, they mutate, they recombine, they do all sorts of things that allow you to see some of the things that the body is doing during behavior, during computation. So we have things that, like what we see in nature, like syncytium, which are large cell bodies with a lot of nuclei in them. So cells fuse together into syncytium, like you see with muscle. You have single cells that have nuclei that can change their shape and their size, and they can change their angle of orientation so that they can migrate and change into different specialized parts. So the orientation, the size of the cells and a tissue type, and all these other things contribute to this sort of morphological computation. Now let's talk about the biophysical embodiment of Moire pattern formation. So these are some cells that are kind of isolated outside of a body. So we want to show how the morphogenesis acts upon individual cells. So this is an array of cells, and I'll show a number of these cell sheets where you have an array of cells that are deformed and turned in different angles and things like that. And so we actually have two sources here. So we have a two-source interference pattern. So the left cell in the sheet is emitting something. It's a source. The nucleus is a source, and it's emitting to both itself and all other cells in this array. So the farther you're away from this left-hand nucleus on the left side of the sheet, the weaker the signal. Simultaneously, you have a light source, and that light source is also emitting some signal, and it has its diffusion pattern as well. And so the farther you're away from that light source, the weaker that signal. But there's this overlap. There are these fringes that overlap here in this region, right about here, and this covers these two cells. They're kind of in the middle, the second from the left-hand side, and the middle cell, where these two signals overlap. And in that space, it provides grist for the mill of morphogenesis, or this interference pattern interactive morphogenesis type effect. And so this is a two-source interference pattern in a single layer of cells and an array of cells, where you have two sources, an environmental source and a genetic source, and they're interacting. So we have this autonomous source, which is biomolecular, and we have this environmental source, which is photic, and they interact and interfere in this zone. And then the zone is where you can have the effects of both that may be amplified. So the interference equals the strength of the interaction. It might be like a behavioral taxis, like we'll talk about later, plus this instructive signal that's autonomous to the cell. This is a little bit more complex way of thinking about this. We might have an external organizer, a source, or a chemical source in the environment, or within the body itself, which is sort of mediating chirality in this cell sheet. So organizer A is mediating chirality that's right-handed, that's going in this sheet on the top, and then the organizer B is mediating left-handed chirality in the sheet on the bottom. Both of these cell sheets sit sort of on top of a substrate, which is stationary. And we have this three-layered structure, one cell sheet displaced from another cell sheet just slightly by maybe half a phase, as you can see. And then we have these different aspects of chirality, which is just the handedness of the sheet. So anything that goes through the sheet, any signal that goes through the sheet from the organizer goes in that direction. So what does this mean for our patterns? Well, this means that we have this, we set up for this interference pattern. We have two offset layers on a substrate, and we have chirality going in different directions. So obviously we're going to get this interference effect, and it's going to affect the pattern formation within both of these sheets. So this idea is taken from this paper, Du et al., Nonlinear Physics of Moire Superlattices. So in materials science, this is in Nature Materials, we have these structures called Moire superlattices. And so there are a lot of interesting nonlinear physics where we can have these different layers of materials that interact and form these Moire patterns. So back to our cell sheet where we have this sort of tri-layered structure with a Moire-like pattern in the center. You can see that we have actually here 3 layers of cells, 2 that are aligned in the same direction and 1 that's offset by half a phase. Those interact, and you can see sort of how they overlap. And then we just take that overlapping section out, and we can see that on the top we have this tri-layered structure with the Moire-like pattern in the center, and then on the bottom we've extracted that pattern out, and that's the pattern that we get. That's the additive component that we get from these 3 sheets. And so we get the structural interference, and we get biomolecular interactions and crosstalk. And we'll get to crosstalk later as well. It's a very interesting phenomenon. So we can actually modify these sheets. So we have our 3 sheets, 2 layers, the top and the bottom layer aligned in the same direction, and then the middle sheet off by half a phase. And so that produces additive patterns. But then we can introduce apoptosis, which means we can kill off cells and kill off their nuclei and change the pattern accordingly. So we have this count for, on the top we don't have any apoptosis, and we get the pattern that we saw on the last slide. We replicate that pattern. In the middle we have 5 cells that are dead, that they get killed through this apoptotic process, and we've changed the pattern. It's now irregular, it's now heterogeneous. And then we also then kill off 7 cells, where we change 7 cells, and we have now an apoptosis of 7, and we end up with an even different pattern. So we can actually intervene and kill off cells. And this is actually what happens a lot in development, where you get cells that die through this apoptotic process. And sometimes that's used in pattern formation, sometimes that's used in the nervous system to refine pathways. And in this case, we can refine our Moire pattern, or maybe more accurately, we can make it nonlinear. So this gives us our 3-layer Moire superstructure. This shows not just how they're overlapping, but it shows the twist angles, the layer separation, and the apoptosis in each layer. So we have 2 cells that die on the top, 1 that die in the middle layer, and then 1 that dies in the bottom layer. In this case, they're all misaligned by half a phase, but I did this to show how these connect, first of all, from nucleus to nucleus, and then secondly, how those the effects of apoptosis, and then how those twist angles can be modified accordingly. So we can see that very clearly here, and this produces a lot of nonlinear effects. So I've hinted at a number of phenomena that involve materials and producing Moire patterns and superstructures of materials. So Moire superstructures are defined on a 6-fold close-packed substrate, and if you do this, you can see patterns that appear as hexagonal modulations. So oftentimes, you can build these materials that have some substrate, and then they have some overlayer, and it's offset to some extent. And if you have these 6-fold closed-packed substrates that have something on top of them, you can modulate their form, you can modulate the surface patterns. So this relies on a symmetry mismatch. So we can model this with a 4-fold simple geometric model, and we can change the morphology of that from stripes to a rhombic network as the twist angle increases. Remember, the twist angle is the sort of offset between the substrate and the top layers. So you might twist one layer versus the substrate, you might bend it in one way versus another, and you can get the symmetry mismatch, and you can get these Moire superstructures that exhibit these sort of changes in patterns, changes in the symmetry across the entire surface, but also you see it exhibits local changes as well. So this paper from Journal of Physics C, the Zhao 2020, talks about honeycombs on square lattices, and they do some geometric studies and strain analysis of these structures at a symmetry mismatched interface. Basically, we have this visual aspect, this imaging aspect, but also a physical aspect as well, mechanical aspect. There are also these phenomena called van der Waals heterobilayers. And these are bilayers that are different in terms of their properties. The composition of van der Waals bilayers are locally identical between layers, but exhibit local variation over long-range distances. So these bilayers, again, they exhibit this twisting, where the twisting action produces this offset angle. So you have two layers, maybe a substrate and some chemical layer on top, or you have two chemical layers, and they twist against each other. So they move in opposite directions, and it produces an offset angle that is sometimes the property of material bonds. So in this case, we're dealing with chemical structures, biochemical structures, and we're able to produce these sort of Moire superlattices. And this is a specific type called van der Waals heterobilayers, and of course, as the name suggests, it can also produce van der Waals forces. We have these topological mosaics, and we also have these Moire profiles that get produced in different types of nanosheets. So we can produce this pattern formation at very small scales. And so this is an example of sort of a spectral analysis of these superlattices. We see Moire patterns and superlattices. We see interference between misaligned and mismatched superlattices. This is a phenomenon in particular called the Hofstadter butterfly. So this is a spectral map of non-interacting electrons in a two-dimensional perpendicular magnetic field. And these kind of Moire patterns exhibit emergent behaviors on a fractal energy landscape. And so there are a number of papers in nanotech, in nanotech, in physics, and in material science that are relevant here. This paper, Moire Patterns and 2D Materials, a review. There's this other paper, The Marvels of Moire Materials, that's Nature Reviews Materials. And then finally, Hofstadter's butterfly and the fractal quantum Hall effect in Moire superlattices, and that's from Nature. That's, I think, where this figure is from. And so we can look at an indirect exciton matter condensate, which is formed by electrons in holes in separated layers, as we saw with the cellular layers that had apoptotic removals. We can have holes in different layers of these materials, and this can yield observed interference dislocations. So we can have the Moire effect in interference patterns by propagating waves. And this gives us this idea of a Moiron, which was described in Herman 2012, which is down here. Periodic overlayers and Moire patterns, theoretical studies of geometric properties, where we have this ruthenium substrate and this graphene overlayer. And if we look at this layer, we see spots that are quasi-periodic hexagonal patterns. So we see that we have these patterns that are kind of like triangles, and then we have these spots that are sort of hexagonal in shape, but you can see the spots. And this is a feature of sort of undulations in this overlayer, in this graphene overlayer, and sometimes removals with respect to the substrate. So we have these different things that can occur when you have a substrate versus an overlayer, and how they're structured and how they interact. We also have this other paper, Universal Classification of Twisted, Strained, and Shared Graphene Moire Superlattices, which discusses some of these patterns and how you can classify them. This is another visualization of Moire superlattices. Again, we have the substrate and this overlayer, and they're the chemical bonds between them, which kind of determine the twist angle, because that's a physical effect of them twisting and changing their configuration. So we have this very specific sort of morphological computation that occurs between the layers. We can calculate all of that out. We can calculate the twist, and then we can characterize the pattern. So this is a link at the bottom here from Exploratorium to Moire Patterns. This paper down here, McGilly, Visualization of Moire Superlattices and Nature Nanotechnology, is where I get these figures from, and it kind of shows sort of the phase angle of some of these patterns and how that can change. Finally, we have this example of this iridium 331 substrate and a graphene overlayer, and we can see this in 3 dimensions, where we have these methods for determining optimal supercell representation of interfaces, and this other paper, Cell Match, combining 2 unit cells into a common supercell, with minimal strain, basically ways that we can calculate using software some of these patterns and some of the ways that the substrates are configured with overlayers and the patterns that they produce. Finally, let's take a look at Moire patterns and behavior. So we've talked about Moire patterns and materials and in development. Now we'll talk about behavior. And in this case, we're trying to train a model to interesting structure, although we're not going to get into the actual training regimen. We'll just talk about how this might be structured. So let's explore the idea of chromotaxis. So this is where we have this Bradenburg vehicle with different sensory organs that are tuned with specific color. And it's going to explore this gradient and be attracted to colors that it can recognize. So this is similar to a phototaxis, where Bradenburg vehicles of a certain type are attracted or repulsed by a light source, and there's a gradient of light. So as it moves towards the light, it gets stronger. As it moves away from the light, it gets weaker, and that affects its behavior. And so we can also think of chromotaxis as sort of like phototaxis, or we can think of it as sort of like a preference. So this is where we have like something like an emotional valence continuum. And this is also something that Bradenburg proposed with respect to Bradenburg vehicles, that you have this emotional valence, that the behavior, how the behavior manifests in the vehicles, and how that can be a continuum that you can explore, much like a chromotactic continuum. Now let's take the idea of Mach bands. So Mach bands are these gradients that they're largely an optical phenomenon. They use them a lot in psychophysics to figure out where people detect changes in that gradient. We have this banding, and there's a gradient from one color gray to another color black, and you have to find the distinctions within them. And so this is one of the classic psychophysics tests, and you can actually implement this in an agent. So in this case, we're going to take some agents, some developmental agents. We're going to give them different viewpoints on a visual gradient, and we're going to train them on this Mach band gradient to see if they can detect differences in that Mach band, or from given different orientations, we're going to detect patterns in this Mach band. And so this involves chemical and physical properties that manifest as collective behavior. So you can have multiple agents kind of explore the same space from different perspectives and produce some sort of collective behavior that is manifest as some chemical or physical phenomena. Then we also have within the agents themselves these physical, chemical, and electrical phenomena, which correspond to its biochemistry and regulatory network, and connectome, which relates directly to behavior. Let's go from psychophysics to thinking more about cybernetic regulation and crosstalks in different types of networks, including work that's been done on genetic regulatory networks. So Mach bands, of course, are these visual illusions, and they act upon cells, neurons, that get tuned to certain inputs, and those cells enact collectively to interpret those fields as outputs. Now, Mach bands can be characterized as a series of luminance plateaus with ramps that define transitions between plateaus. So they're visual phenomena, they're visual illusions, but they can also be physical phenomena that have actual values as opposed to being a purely visual phenomena or a mental phenomenon. Mach bands result in illusory light and dark bands, or stable regions and boundaries between regions. So this is essentially something like what you see in morphogenetic gradients, where you have these differences across the continuum, across the gradient, and there's stable regions and they're unstable regions. And people who've studied Mach bands have found that you can characterize them with a number of models, including feature-based models, rule-based models, and filling-in models. And those are all things that we propose at the internal mechanism where the brain does to interpret those Mach bands. But remember, Mach bands aren't just visual phenomena, or aren't just visual illusions, they're also physical, hence the term psychophysics. And so we can link this to physical phenomena as well, physical gradients. And so We can do this by looking at mock bands with relation not only to morphogenesis, but also to collective behavior. So this is where we have this pattern formation in the psychophysical domain. We have some agent that's observing these mock bands, and they're summarizing these mock bands as in a spatially explicit rule-based model. So they're thinking about, or they're sort of interpreting mock bands as something with some sort of spatial resolution, and they're using rules to interpret what, the different conditions of those mock bands, where the boundaries are, where the stable regions are, and so forth. Now, the observers, of course, have two different ways that these lessons can be expressed. The first is through morphogenesis. So an agent can sense and learn morphogenesis. So an agent can observe morphogenesis in one agent and replicate it in themselves, or they can learn morphogenesis and learn things about their adversary if it's like a predator observing prey or something like that. So you can have multiple ways that morphogenesis can be transmitted through observing mock bands or some other pattern in nature. Another way that mock bands can be manifest is through collective behavior, and I think that diagram kind of showed how you can arrange a population of, or how you can configure a population of agents to observe the mock bands, to observe that gradient from different perspectives, and gain collective information that can then translate into collective behavior. So this is especially useful not just in mock band observations, but in the observations of swarms and flocks, and learning something about their internal structure from that. And then getting to work in genetic regulatory networks, we've discovered this idea of crosstalk, and people also use the term crosstalk in the neuroscience literature. But this is a paper on talking about crosstalk in gene regulatory networks and how gene regulatory networks that operate in parallel can exchange noise and information. So crosstalk is where a signal transmitted in one channel, in this case channel A, can have some sort of undesired effect in a parallel channel or channel B. And so this is, a diagram of closed loop feedback, closed loop second order feedback for both channel A and channel B, but that there's some contribution from channel A that gets transposed onto channel B, and so this provides some sort of noise to channel B, although sometimes this noise is not always deleterious, sometimes it's beneficial. So sometimes crosstalk can be good as well as bad. And in this paper, they talk about it mainly as being bad and something that needs to be controlled, but sometimes crosstalk can be good. Sometimes crosstalk can accurately help, or it can help agents accurately interpret environmental signals. And sometimes you can have compensatory crosstalk where channel A can assist channel B and vice versa in detecting patterns, or if this is a scenario where we have two pathways in a connectome where those kind of give each other information. Say we have two sensors on the front of a vehicle, we have two channels in parallel, and then there's selective crosstalk that exchanges information. Sometimes if you see sometimes if you think about mammalian nervous systems or bilateral nervous systems, you have crosstalk between the right side and the left side of the nervous system. And that can be sort of this mechanism. So thank you for your attention. There's a link to the phasic interest group and some of the work we've been doing, and then a link to the Developmental Neurosimulation initiative, which is within the Representational Brains and Phenotypes interest group, and some of our publications and talks there. Thank you.